So welcome to this tutorial video. So before we even begin looking at our topic for today, make sure that you subscribe to this channel and make sure that you hit that like button. So my name is Tembo from Step Up Academy, and in today's tutorial video we'll look at a topic that we call glycolysis in biochemistry. So I believe most of you have come across this topic, but for those that haven't come across this topic, don't worry. As we conclude this session, we'll make sure that you understand. So, glycolysis. So just to give you a recap, I believe in your biochemistry sessions, you've looked at the four important macromolecules. For example, you've looked at carbohydrates, you've looked at proteins, lipids, amino acids. So you've looked at those things. Now, when you talk about carbohydrates, you've always been told to say carbohydrates, these, are molecules that are used for energy production. They provide the body with energy. So, as somebody who is studying health-related courses, you have to understand to say how is this energy that we're talking about produced. So, when talking about carbohydrates, when they are broken down, they're supposed to produce energy at the end of the day. And the first process that is involved is what we call glycolysis. So this is what we are looking at today. We want to see to say when you eat your carbohydrate meal, how do they provide you with energy? And we are saying that the first process that is involved in that energy production is what we call glycolysis. So you need to understand this because there are various, cells in the body that highly depend on glycolysis for them to function normally. For example, the brain cells, they depend on glycolysis to provide them with the energy for them to function. Okay? You can talk about your brain cells, you can talk about your muscles, all right? All those depend on glycolysis. Even the red blood cells, for them to be able to be kept in that concave shape, they need glycolysis. So, just to make the term easier for you, you can see that this is glycolysis. If you break this word down, you have got glyco here, that is just coming from the word glucose. Then lysis just simply means breaking down. So when you put these terms together, you understand that glycolysis is basically the breakdown of a glucose molecule in order to produce pyruvate. So, as we conclude, you should be able to understand that the overall idea behind glycolysis is that you are supposed to break down a glucose molecule that you've obtained from your carbohydrate meal in order to produce what? Pyruvate. So, you can see a picture of bread here. So what I'm just trying to remind you by this picture is to remind you to say what you are breaking down is a glucose molecule that you've obtained from your carbohydrate meal. So there's somebody here who has eaten their bread in the morning for their breakfast. So how does now that bread provide them with energy? How do they, manage to be energetic throughout the day because they've eaten their breakfast? So what we are saying is that from that bread they've eaten, we've obtained a glucose molecule. But in glycolysis, what we want to do is that we want to break down that glucose molecule and we form a molecule that we call pyruvate. That is what glycolysis is all about, breaking down glucose and forming a molecule that we call pyruvate. So you can see here to say this pyruvate can later form what we call citrate. That's just in advanced stages such as the Krebs cycle, which we're going to look at in the next session. Then that citrate now can undergo further reactions and produce ATP molecules, that is in other advanced stages. But in this session, what I just want you to understand is that you're just breaking down a glucose molecule in order to produce pyruvate. So the definition here, we have it just like I had said in the introduction to say, glycolysis basically is the process which involves the breakdown of glucose in order to produce a pyruvate molecule. So you and I know very well what to say. Glucose is a 6-carbon atom molecule. It contains 6 carbon atoms. Then this molecule that we're producing, which we call pyruvate, has got what? 3 carbon atoms in case they ask you that question. So what is the general overview of this topic? We are saying that glycolysis is irreversible. So when the process starts, meaning it can't go back. And you should know that this glycolysis takes place in the cytoplasm. So I believe you know how the normal animal or human cell looks like. Then in the cytoplasm, that's where glycolysis takes place. What you are just seeing down here is basically just the overall equation that shows you how this reaction takes place. So you can see from your left here, you've got a glucose molecule, okay, plus these other molecules like you've got 2 phosphate groups there, you've got 2 ADP molecules, you've got 2 NAD molecules. Now, from these, you should be able to produce pyruvate, 2 molecules of ATP, NADH, 2 hydrogen atoms, as well as 2 molecules of water. But what I just want you to understand is that from a glucose molecule here on your left, you should be able to produce pyruvate. Then if you understand that, then you've understood glycolysis. Now, if you look at this picture very well, you can see from your right to from your left to say this is basically a glucose molecule. In as much as they have told us to say this is a glucose 6-phosphate, I've put this on purpose to say glucose 6-phosphate. I'll show you what is the importance of this glucose 6-phosphate soon. But what I just want you to understand is that what you are seeing on top, okay, on this left part, is basically the structure of a glucose molecule. Now, what you are seeing on your right is what we call pyruvate. Remember, we said in glycolysis, what we're just doing is that we are breaking down this glucose molecule and then we form a pyruvate molecule. So pyruvate molecule that we are forming, you can see, if you count, it has got 3 carbon atoms. 1, 2, and 3 there. It has got a carbonyl group. It has got a carboxyl group here, a carbonyl group, and a methyl group. So let's make progress. Now, glycolysis involves 10 steps. Glycolysis involves 10 stages. So for the sake of exams, you have to understand that these stages are divided into 2. We've got the first 5 stages that we call the preparatory stages or the investing stages. Then you've got the last 5 stages which we call the harvesting or yielding stages. Now, you may be asking yourself to say, why is he saying to say the first 5 stages are called preparatory stages or investing stages? So what happens basically is that in the first 5 stages, you're going to utilize 2 ATP molecules in glycolysis. You're going to use them. No wonder we call them investing, because ATP is an energy molecule. So in the first 5 stages, we're going to use 2 ATP molecules. Now, in the last 5 stages, we're supposed to harvest those 2 ATP molecules that we used. That is why when you looked at that equation that I'd shown you there, you had seen to say in the first, reaction, you're going to utilize ATP, and then as you go to the right of the equation, you find that you're also producing 2 molecules of ATP. That's what I want you to understand. Now, in these 10 stages that I'm talking about, what I just want you to know are specific parts that I'm going to emphasize on. These parts which are highly examinable. So you can see that you've got a glucose molecule there on top that you've obtained from your carbohydrate meal. Then for it to have energy, it's supposed to be broken down. And we have said that the process that is firstly done for this energy to be produced is glycolysis. So this glucose molecule is going to be broken down in these 10 stages, and then at the end of the day, we're supposed to produce pyruvate, which is down there. So we're going to go through these stages, and I'll show you and emphasize on which parts you have to understand the most. So everything that that has to do with glycolysis is based on this picture. So make sure that you follow through and you understand. So let's start with the stages. So we've been taught to say we've got a glucose molecule that is on top here. Now in glycolysis, we want to break it down so that we produce pyruvate down there. So this is what we're talking about. You can see here to say from glucose, we're supposed to produce pyruvate down there. So you've got a glucose molecule on top there. Now this glucose molecule is going to be broken down into what we call glucose 6-phosphate. And this process involves ATP. Remember, I had shown you a picture there which showed glucose 6-phosphate. So I'm going to take you back so that you just understand what we mean when we say glucose being broken down to glucose 6-phosphate. What you've just done is that you've added a phosphate group at carbon atom number 6. So that's why this picture was important. So if you count here the carbon atoms, you've got 1, 2, 3, 4, you've got 5 there and 6. So you can see that at carbon atom number 6, they have added what? A phosphate group. So basically that's what we mean when we say glucose 6-phosphate. If they were to add it at carbon atom number 3, then that molecule become glucose 3-phosphate. If that is if they add a phosphate group on carbon atom number what? Number 3. So you can see here to say glucose has been converted to glucose 6-phosphate by utilizing this ATP molecule. This is in the first 5 stages. So you've utilized an ATP molecule at this particular stage. Then this stage is also catalyzed by an enzyme that we call hexokinase or glucokinase. So there are 2 enzymes here. You may wonder to say, why are there 2 enzymes? Now, these 2 enzymes perform the same function, but they are used differently. I'll start with the glucokinase. So glucokinase is only used when there are high levels of glucose in your bloodstream. Then the body is going to utilize glucokinase because it's very fast, so that it reduces the number of glucose content in your blood and take it back to normal. But under normal circumstances, it uses hexokinase. I repeat, your body is going to utilize glucokinase to convert glucose to glucose 6-phosphate if your blood sugar levels are very high. But if everything is under normal circumstances, it's going to utilize hexokinase to convert glucose to glucose 6-phosphate. And I've said this process involves the utilization of what? An ATP molecule. Let's make progress. So, glucose has been converted to glucose 6-phosphate. The next step is converting glucose 6-phosphate into what we call fructose 6-phosphate. Now, this process basically involves an enzyme that we call glucose 6-phosphate isomerase. Because you and I know very well to say fructose is basically an isomer of what? Glucose. So, from glucose, we've converted it to glucose 6-phosphate. Glucose 6-phosphate, we've converted it to what we call fructose 6-phosphate. So I can see that you can see on your screen to say there is this equation, okay, or there is this pathway that is going to the right. Then there is this pathway that is coming down. But for now, I just want you to focus on this pathway which is what? Coming down here. So, fructose 6-phosphate can now be converted into what we call fructose 1,6 bisphosphate. So it is the same concept. When they say fructose 1,6 bisphosphate, what they mean is that you've just added a phosphate group at carbon atom number 6 of this fructose molecule and carbon atom number what? 1. And we are calling it fructose 1,6 bisphosphate. Now, you can also see that in this stage again, we are utilizing another ATP molecule. So that is what that's why we said to say these stages, the first 5 stages involves investing. So you are investing energy because you are using ATP. But you realize that as we make progress, as we conclude in the last 5 stages, we are going to produce our ATP that we used. So, recap. Fructose 6-phosphate has been converted to what we call fructose 1,6 bisphosphate. Now, there is a very, very important enzyme here that we call phosphofructokinase 1. This is the rate-limiting enzyme of glycolysis that you must know. Fructose 6-phosphate is converted to fructose 1,6 bisphosphate by the enzyme that we call phosphofructokinase 1. And this is the rate-limiting enzyme of glycolysis. It is the most important enzyme of what? Glycolysis. Now, you can see here to say ATP and citrate, they inhibit phosphofructokinase 1. In case you're asked a question to say, what are the regulators of what? Glycolysis. So, your answer should be ATP and citrate, as well as AMP. So, I'll talk about ATP and citrate first. So, ATP, why does ATP inhibit phosphofructokinase 1? The answer is very simple. Since when you're breaking down a glucose molecule, you want to produce pyruvate at the end of the day. Then, if you already have ATP, then why should you break down a glucose molecule, because ATP is already an energy molecule, so the body will not need ATP. It's the same with citrate. Citrate is a molecule that is involved in Krebs cycle. Like I had said, after we break down this glucose, we form pyruvate. Later on, we can form citrate, which can now proceed to other process that can form now numerous number of ATP. So if you already have citrate, then your body will not need glucose, because citrate can form energy at advanced stages. AMP. AMP simply means adenosine monophosphate. So we are saying that AMP is going to produce or is going to promote phosphofructokinase 1. Why? It's because adenosine monophosphate is not ATP. The body can't use it for energy. So when the body detects to say there is high numbers of AMP in the bloodstream, they are going to say, I think this is the time that we need glycolysis, because what we want as is ATP at the end of the day. So, recap. Fructose 6-phosphate has been converted to fructose 1,6 bisphosphate. What enzyme are we using? Phosphofructokinase 1. What can inhibit it? ATP or citrate. What can promote it? AMP, adenosine monophosphate. So, let's come back now to the right hand side to say, because you can see that there's another pathway, okay, that is coming this side, but it's heading the same direction down. So, you can see that fructose 6-phosphate can also be converted to what we call fructose 2,6 bisphosphate. But as a medical personnel, you have to understand under what circumstances. So, fructose 6-phosphate, this molecule can go in this direction if the glucose levels are high. So why does the body choose to go in this direction? Here is the answer. When the glucose levels are high, the body wants to maintain what we call euglycemia or normal glucose levels. So what it will do is that it's going to speed up this process of glycolysis. And the only way it can do so is by using this direction as an alternative. So it's going to convert fructose 6-phosphate into what we call fructose 2,6 bisphosphate using the enzyme that we call phosphofructokinase 2. You saw that in this reaction we used phosphofructokinase 1, but in this direction the body is converting fructose 6-phosphate to what we call fructose 2,6 bisphosphate using phosphofructokinase 2. Now you can see here to say there are other regulators of glycolysis such as what? Glucagon and insulin. So you have to understand to say under what circumstance does the body under what circumstances does the body utilize insulin to promote this process. So you and I know very well to say glucagon and insulin, they regulate sugar levels in the body. Now what glucagon does is that it converts glycogen back to glucose. Now if you ask yourself to say, but what I'm trying to do here is I'm trying to break down glucose, but this glucagon goes in reverse. Its overall goal is to form glucose. No wonder it's going to inhibit this process, because glucagon promotes their production of glucose. But what we are doing as here, we want to break down glucose. No wonder glucagon is going to inhibit this enzyme. Now, insulin. Insulin promotes the breakdown of glucose. Remember, when we got diabetic patients, you give them insulin so that you can reduce the number of glucose levels in the bloodstream. So, it's the same thing that we're trying to do here. We're trying to break down this glucose in the blood so that we can form pyruvate. So insulin is going to promote this process, or it's going to promote this process by promoting phosphofructokinase 2. Now what will happen now thereafter? So this fructose 2,6 bisphosphate, okay, can now promote phosphofructokinase 1. Remember I said this is a rate limiting enzyme, meaning this stage is very slow. Okay, this stage is very slow. So the body, if the body senses to say, the number of glucose levels are very high, then it's going to use this pathway. If it uses this pathway, now this molecule that is producing here, what we call fructose 2,6 bisphosphate, is going to promote the production of phosphofructokinase 1, which now speeds up this process, which is the conversion of fructose 6-phosphate into fructose 1,6 bisphosphate. So, from that stage now, let's make progress. You have seen that in the first 5 stages, we have utilized only 2 ATP molecules. That's at this stage and at this stage. Now, what happens to fructose 1,6 bisphosphate? Now, this fructose 1,6 bisphosphate is split into 2 molecules. So the first molecule is what we call glycolaldehyde 3-phosphate. Glycolaldehyde 3-phosphate. Then it's also split into another molecule which we call dihydroxyacetone phosphate. So, I repeat, when the body forms fructose 6-phosphate to fructose 1,6 bisphosphate, this fructose 1,6 bisphosphate is split into 2 molecules. The first molecule is what we call glycolaldehyde 3-phosphate. The other molecule is what we call dihydroxyacetone phosphate. So, from this stage, the process must continue until we produce what? Pyruvate at the end of this process. Now, they may ask you a question here to say, what happens when this dihydroacetone and glycolaldehyde 3-phosphate are produced? I must make mention to say glycolaldehyde 3-phosphate is the molecule that has got a high probability of proceeding to the other stages up to the 10th stage. However, dihydroacetone, it has got less probability of producing to the next stages. So what happens is that there is an interconversion of dihydroxyacetone to glycolaldehyde 3-phosphate, because this one, it has got a high chance of proceeding to the other stages. That you must know. Now, the other stages which are in between here, from fructose 1,6 bisphosphate until you form phosphoenolpyruvate, but most of them are not important. For the sake of exams, I'd put here to say what you have here, 7, 7 simply means this is the 7th stage. Remember we said glycolysis has got what? 10 stages. So, at stage number 7, we've got a molecule that we call 1,3 bisphosphoglycerate. So this molecule now is the one that forms the first ATP in the harvesting stage. Remember, in the first 5 stages we invested ATP at this point and at this point. Now, in the last 5 stages now, we want to harvest or produce our ATP. And I've said they can ask you to say at which stage or which molecule produces the first ATP when you're harvesting in the last 5 stages. I've said it's at stage number 7, and this molecule is called 1,3 bisphosphoglycerate. So the process continues. So from there, you're going to go into the last stage. Okay? There are other stages there that I'm just jumping because they're not very important. Then this one, when you're converting phosphoenolpyruvate to pyruvate, okay, just at my screen as cut, just at the end of this stage, from phosphoenolpyruvate, we're producing pyruvate. That is the last stage. So this is the last stage that we're talking about, where we've got phosphoenolpyruvate being converted to pyruvate. What enzyme do we use? We use an enzyme that we call pyruvate kinase. And at this particular stage, and I must make mention to say this is the last and the 10th stage. This we also, at this particular stage, we also produce our last ATP. And the enzyme that we use is what we call pyruvate kinase. So you can see very well to say phosphoenolpyruvate, that is here, is being converted to pyruvate. This is at the last stage or 10th stage, using the enzyme that we call pyruvate kinase. And at this particular stage again, you produce your last ATP. You can see on the right to say ATP and citrate also inhibit pyruvate kinase enzyme. The concept is the same. What the body is trying to produce is ATP, alright, after the glycolysis is done. But if ATP is already there, if citrate is already there, then there's no need of the body undergoing glycolysis. So a recap of everything. Glucose has been converted to glucose 6-phosphate using these 2 enzymes. Then glucose 6-phosphate has been converted to fructose 6-phosphate using what? Glucose 6-phosphate isomerase enzyme. Then fructose 6-phosphate has been converted to fructose 1,6 bisphosphate, okay, using what? The rate limiting enzyme, the most important enzyme, phosphofructokinase 1. And we've said that this fructose 1,6 bisphosphate will now be split into 2 molecules, which we call glycolaldehyde 3-phosphate and dihydroacetone phosphate. And we've said glycolaldehyde 3-phosphate is the one that has got a high chance of proceeding to the last stage. And I've said that at stage number 7, they can ask you to say, at what particular point do you form your first ATP in the harvesting stage? I've said it's a stage number 7. And the molecule that we use is 1,3 bisphosphoglycolate. Then the last stage that we're talking about is the conversion of phosphoenolpyruvate, that is down here, to pyruvate. It's just that my screen has cut. The last molecule down is supposed to be pyruvate. So this is the last stage, and it involves the enzyme that we call pyruvate kinase. And in this last stage, we are also producing ATP. So, 3 important points that you have to understand from glycolysis. There are 3 irreversible reactions. The first one is the first reaction that is catalyzed by hexokinase or glucokinase. So that reaction is reversible. This reaction here, glucose to glucose 6-phosphate, is irreversible. The other reaction that you have to know is what we call the third reaction. That is catalyzed by what? Phosphofructokinase 1. This reaction, where fructose 6-phosphate is being converted to fructose 1,6 bisphosphate. This stage is also irreversible. Then the last stage, like I had said, the last stage involves phosphoenolpyruvate, that is down here, being converted to what? Pyruvate. So the 10th stage is catalyzed by what we call pyruvate kinase. Okay? So this is the last stage, where you're converting phosphoenolpyruvate to pyruvate. Then by that you can conclude to say you've broken down your glucose in these 10 stages and you formed your pyruvate at the end of the day. Now, you must know that the most important enzyme of glycolysis is what we call phosphofructokinase 1. It's the most control point in glycolysis. So these are just some questions that you can go through to see if you understand this tutorial. Thank you so much. Make sure that you subscribe to this channel so that you watch or understand other topics that we're going to talk about in different courses. Thank you.